welcome back to the lab. Welcome back to the lab of class 3. And so I've opened my R already. So you can essentially see already that I have opened the RMD file that is provided with the class. And so those of you that have seen the class already, so there was the question about the Stargazer printout, that didn't happen because type was LaTeX and type should have been text. So changing type to text will make the whole thing work. So again, please copy the RMD file into the lab folder in your class 3 folder and run the entire code. This will give you a PDF that will contain, you may need to change the init. If it's still changed to HTML, you may need to change it to PDF. A little background story with that is that you may have to install MikTeX, which is a LaTeX application, which may be required by your RStudio. It should not be a problem to install this. If you have any problems, please let me know. But I think it's quite straightforward. If you give it some thought and read up a bit on it, it should not be a bigger problem. So in any case, so we are now looking at lab 3, and we will be talking. So please study the PDF file, which is very helpful essentially for you to review what is being asked from you for lab 3. So if we look here at the assignment, so we're looking at lab 3 R code. We have that. We look at different seeds, and just as a background story, and we have here. Okay, so it has finished already. So now we can open this. So I'm opening the PDF file that was generated from the lab. So essentially we have here, we're simulating data now. So we are looking now to understand the influence of unobserved confounders. For this very reason, we will create simulation studies. We will create random variables with known population parameters. So for that very reason, we will be using the same randomization scheme, this randomization of parameters of variables that essentially were done by Professor Sang for the lecture. The only difference is here the set seed function. So the set seed function, it's a little bit hard to grasp, but what it essentially means is that R will be computing not really randomized numbers, but something like pseudo-randomized numbers. So these will follow a certain pattern and need one initiating value. And this initiating value essentially is absolutely irrelevant, the magnitude, what it actually is, but it is needed to create a starting point for that algorithm that R will be using moving forward to randomize its numbers. And this randomization will then still be presenting itself in the same way and fashion as it is designed here. So R norm, for example, is a replica of 100 times with a mean of 35 and a standard deviation of 5. Our binom will still be a replication of 100 times, which is either a 1 or a 0 with a mean of 0.5. And the relationships will still be the same. So if you do here a mean 50 plus 0.01 age minus 0.01 multiplied by 6, you essentially, you get still the same kind of simulated data set as you would have gotten with a seed of 1 if you put a seed of 10. But the starting point is a different one. And by that, you essentially, you make it shareable and reproducible, meaning if you take the same seed as a starting point, you will get always the same kind of sequence of simulated numbers if you replicate it. So this being said, R by default would do it with random numbers. If we are doing it here in an automatized shape of form, we essentially give it a certain kind of different outline. And essentially we will now essentially allow for it to take different shapes and forms. And by that, essentially, we can customize not only the seeds, but we can also customize the confounders. And this will go hand in hand with what we're supposed to be doing for the lab. So let's walk through this PDF. So this is the seed. This is the seed function. So if we look now at this set seed command, we're essentially getting here a certain kind of simulated data set. And if we fit the linear model through that, we are essentially getting a linear function, which we know already, right? So now we can verify the effect of the constant 1 in the R formula. We use the model matrix function to obtain design metrics that R uses to calculate regression estimates. So if we are now, and this is always a good point, to compare the model matrix per se. So the model matrix essentially is, it's a little tough to explain, but essentially it's an internal kind of process that means, I'm not really sure why Marcel and Bino have included it here, but essentially kind of it goes into the background of the estimation of how these coefficients are basically estimated based on the data. And the idea of this was really to make the case that the model matrix is going to be the same when it includes the 1 plus versus not includes the 1 plus. And so you have here for each individual, you have this entire matrix lined up. And you have the exact identical aspect for the model when you don't include the 1 plus. So this is not really essential to the lab. It's just something more like an FYI. So the 1 plus really just serves for the purpose to make it mathematically correct, because it represents the intercept and the mathematical equation, but I don't think it's actually, so it's not necessary to include it in the actual R code. So from that end, I would see it as a lot of low priority to look at this in more detail. Okay, so now we're adjusting for the confounder age, which essentially is done in the same fashion as we have done it in the lecture. I also want to make sure that you know how to extract coefficients as well as confidence intervals. So with coef fit2, you essentially get the coefficients out of the model, which allows you the extraction of intercept, of blood glucose and age. Whereas with confidence intervals, you essentially get the estimates for sugar levels as well as age, and allow you to kind of determine whether this is a significant coefficient or not. With the Stargazer package, you can depict both of your, and here was the text type. So essentially here you can actually see the output. So you have here a characterization of both models next to each other. So it's a very nice and neat way of presenting this data. So then lastly, we're also going to talk about loops. So we have I equals 1 to 3. So we have now 1, 2, and 3 in I. So we can now run a for loop. So if you pack a certain number of a sequence of numbers into I, and you are initiating a for loop, you will have a repetition of whatever you add into the curly brackets. So here we have, if you look at this here, so you have 4 I in 1 to 3. So 1 to 3 with the colon separated by the colon gives you 1, 2, 3. So if you are assigning I in 1, 2, 3, you get exactly that. So if you run this for loop in 1, 2, 3, 1, 2, 3, so 1, 2, 3. You get a repetition of as often as this number in I is being, so it repeats it by the length of I, and it repeats it assuming that I is 1, 2, and 3 for the first run, second run, third run. And if you print I, which is done here, it basically gives you exactly first run, it prints a 1, for second run, it prints a 2, for third run, it prints a 3. Allocate and fill method. The recommended method for writing a for loop is to allocate and fill method. It provides an sufficient use of memory rather than a copy and append method. So that means that essentially you want to have a vector mode. So you have a fill vector I, where you basically have I plus 1. So the fill vector is in essence nothing else. So you have a fill vector of I. So when you run this from 1 to 10, you essentially, what is being meant is that when you have okay, that's a tough one to explain. Just visually. So essentially, so let's think of you wanting to build a vector which has the length of 10, and with each run, with each iteration of your loop, you essentially have a calculation, and you want to have that output of that calculation being added or put for that specific spot of the iteration into your object. You would actually do that by basically adding this with each run. So you can either start with a vector of length 1 or length 0, and then add 1, then 2, then 3, then 4. And so you're appending, so to say. You're appending to your vector, and you make your vector longer and longer and longer. What Marcel and Pinot are here advocating is to essentially use an overriding allocate and fill method. So you make a fill vector of zeros, and then you basically overwrite those vectors, those vector spots, with the respective result of the calculation, which is here an arbitrary I plus 1. and you basically overwrite those zeros with this I+1. And by that, you are nicer to your memory, and you make a more efficient use of your memory. If you would copy and append, this would use more resources. So now this is obviously at this magnitude we're looking at right now. This is not really a conversation piece, but yeah, it may be a conversation piece when you essentially have really large data sets. So from that end, I would very much keep that in mind. And this is actually very good input. A more generalized function is essentially here repeating the same expression with random number generation expression of any language object, part of the speech expression. We essentially can do the same thing with replicate. So if we want to quantify set seed here, again, indicates the start of this pseudo randomized number sequence. And if we want to do this here with 6 different replication of the very same data set, so this is R norm, this is n of 10, around the mean of 0 with a standard deviation of 1. So this will essentially give us 6 different vectors of the length of 10, which essentially will give us a matrix of length of 6. So, and this is essentially what is being advocated here as an alternative to a for loop. And it's a possibility. It's a possibility. Hypothetically, I would stick with the for loop because it gives you a little bit more wiggle room, a little bit more flexibility, and allows for more. I have also bluntly, I have to say, I have not really done much research on replicate and the advantages of a replicate. So I personally would think that it would make more sense to kind of go with the for loop, but that's just me. Okay, so now the lab exercise is essentially set different seeds and see how the results changes, particularly coefficient for the blood sugar level. So essentially we have seen now the quantification of the blood sugar level, so we can just go up. We take that very we are taking this code. So now essentially we have set seed of 1. We have seen that result, so let's do 4, and let's see how it compares to the 1. Oh, we do ln, the fit. Fit is ln of SPB as a function of SL plus age. So now the summary ln fit, summary fit, summary fit. So here essentially we see now it's clearly a different result than the one we've seen before, right? So, we're seeing if we're doing this now with 8, we're seeing a whole different seed than 8. So if you run this down, again, different results. So if you do, you could hypothetically, and I think this would be nice to put in a for loop. So if we are now, and this is the last example, how we actually can then compare our 4 and 8. So let's do fit 4 and fit 8. Let's leave that as our examples for the calculation of, oops, the replication. Okay, so if you run this now with 4 and 8, So we see here substantially different results. Okay. And these are different. And we have seen that already. So choose a different magnitude of confounding. What does that even mean? Okay, so here we see definitely age. We see differences in terms of confounding, which are basically here presented by differences in our associations, right? So we have here differences, for example, and associations in terms of age has a certain influence, our binom has a certain influence, size has a certain influence. All these have different influences. If we want to change now some of the influences on our blood pressure, and because blood pressure here, if you look at this, so basically the blood pressure here is given with a certain mean, with a certain association with serum glucose, with a certain association here with age and a certain association with sex. If we want to manipulate this now, the way to manipulate this is now to essentially use a different estimate. So if we say here, this is like our beta 1, we're changing this 2 with a 1.5, and we're changing age influence by, let's say, a beta 2, by, let's say, a 1, and our, okay, then we can also change standard deviation. So here, this SD, this can be changed by SDV equals, can be 10. And we can also change my seed. Now again, back to 1. And this can be, so this is the beauty. So in coding, you speak for this kind of coding, you would refer to this as quote unquote hard coding. If you are packing this into an object and you're only using this object to refer to in your actual code, in your actual model, this would be called quote unquote soft coding. So now we are soft coding now. Better too. We are soft coding now our data. So this is not changing the standard deviation of this to SDV and my seed. And we're going to change the seed now to my seed. So now this is our new model, right? And this looks different than the initial one. So you can do this now quite elegantly. So you could do this now quite efficiently. So we changed now. For the lab exercise, so we have changed now. Magnitude of confounding, which is our betas. We have refitted the model, the change of SL to SPP. We have now changed here magnitudes of error. We have changed the standard deviation, the my seed. So all of this can be changed. So if you want to compare these models now, you would need to know, you would need to now run this data now in a larger effect. So write a for loop to gather results from 1,000 seeds and quantify the percent bias of estimating coefficient of SL due to omitting confounders. So what does this mean? So we essentially trying the following now. Okay, so this is a little bit more involved. So what we need to do now, and I will post this code actually. I think this is a little too tough for a starting session. So what I want to do, so we want to do beta1 true, which is our actual beta that we have been working with before. So this is our beta1. This is the association between blood glucose or serum glucose and blood pressure. So now we are essentially, we're going back to this guy, to this association. We take this all the way down. So we have now a beta1 true. This beta1 true is 2. We are now essentially looping through our seeds. So we're gonna do num reps. We're gonna do this 1,000 times. We're gonna do 1,000 different seeds, and we're getting a coefficient of SL. So we're gonna get num reps is 1,000. And we remember it's fill, overridden fill. So we do a beta1 dot CA, which is vector numeric length equals num reps. So I will post this code actually. This is a little bit too rough for a second session. Okay, so now we have set my seed. My seed will now be essentially chosen Okay, so here's a new function, which is SQL length. So SQL length num reps will essentially give you a sequence of 1,000. So essentially, it's the same as 1 to 1,000. And so this is a comment for this. So I'm looking at Marcel Albino's code, and there's some. So my seed, which we have said is now set seed, is now in 1 to 1,000, or alternatively, we can also do SQL length. So I'm just preparing you for that, because the answer key would be what SQL length of num reps will be the same thing, right? I'm hashtagging this one out. So for 1 in 1,000, we're essentially getting now different set seeds, and this will get us to different summary fits. So this will essentially now run through, take set seeds from 1 to 1,000, will accordingly use different beta ones, and we're gonna do this beta1 true, that's 2. And we will now make this again 0.5, like in the initial form. This is going to be 5. Standard deviation is 5 again. Okay. All right, good. So this fit is now SPP SL plus H. Summary fit is going to be this, and now we get a beta1 dot CA which follows now my seed. So this will now take the respective spot in the for loop. So we are running this from 1 to 1,000. So we get now my seed with each iteration, we get a number from a 1 to 1,000. So beta 1 will essentially now fill this coefficient that we're extracting now. So we get a coefficient. Where's it gonna write it correctly? We're getting coefficients now, coefficients from my fit. No, it's actually fit, not my fit, which is SL. So what this code will do now is to calculate now 1,000 models and will extract these coefficients out of each model. So these are all the coefficients of the various models with set seeds from a 1 to 1,000. And why are we doing this? Because we want to have the relationship between beta 1 true and beta 1 CA. So if we are getting now a mean of this beta 1 CA minus the beta 1 true, beta 1 dot true, which we should have up there, and quantify the percent due to omitting confounders, percent bias. Okay. So now we can actually, what we can do now is, okay, we can, yeah. So we can make a little difference on bias. We can quantify better bias by quantifying beta 1 CA minus beta 1 true divided by beta 1 true. What is that? What is this? So beta bias, this is this one. This is now the fraction of that. Multiplying that by 100 makes it a percentage. And depicting that now, we get us the percentage difference, the percentage bias. Continuing on this aspect, we'll divide beta 1 true. This will do the mean. That's the mean percentage multiplied by 100. Gives us a mean percentage, mean bias on minus 0.20 with 1,000 different seeds. So this is the mean difference. Okay. So I know that was a little heavy. So okay, let's walk around this. I actually had to kind of like take a deep breath here. So I should have probably prepared more for that lab. Okay, so once again, let's recap this whole thing. So we have here set seed is the starting point for a pseudo randomization, a pseudo randomized sequence. Using this with our norm, our B norm, which is randomized normal distribution, randomized binomial distribution, allows for the generation of random variables according to means or means or approximated means for the binomial distribution that are kind of following your selection. Using this in a fit will give you associations according to those associations that you have entered in your randomization generating code. So now, if you, then here's the model matrix, which essentially shows the consistency between 1 plus serum glucose and serum glucose. I am not sure how that found its way into the lab. I'm not sure whether that is relevant. But yeah, essentially, so 1 plus serum glucose and serum glucose is consistent, and the way why you would put the 1 plus serum glucose is to give it the same layout as you would have in a mathematical expression of your linear regression model. So FIT2 is a multivariable model. You can extract coefficients and confidence intervals from your models. This is true for all covariates that you have included. And you can also depict those automatically using the Stargazer model. So you have here with Stargazer, you have a million different options, and you can depict it in a way that will allow for you to extract all parameters as you want them. The loops are basically iterative processes following the 4. You see from I to 1 and 3, you can actually see each being run independently. You see the print I will show you each number that is assigned to there. That doesn't mean it really needs to be 1 to 3. And I just want to also show you this. You can hypothetically also do this, which is nice, which is C2, 4, 6. Then I will basically do this. So you can basically everything that generates length for you, or a sequence that is being assigned to I, it will run it as long as I is, and it will run it by I grabbing whatever numerical value you throw at it. So if you have 2, 4, 6, it will take 2, 4, 6, and it will run 2, 4, 6 through it. The allocate and fill method, yes. So you don't append. So if you have a numerical simulation like we just did, where you create 1,000 entries, it's more efficient for your memory and for your processor to essentially do a fill vector. So to fill, so you create a vector of 1,000, of a length of 1,000, and you give 0 for each of these entries, for each of these spots, and then you override each of them. This is a better approach than appending. So starting with 0 and then appending one after another. So this is just more efficient. That's this. Then the replicate function, which I personally don't use. I prefer 4. These are the differences between the seeds. When you use different seeds, you have different starting points for this pseudo randomization. And then you go further. So here we can vary, and this is how you can vary between your randomizations. This will give you different words. So please, I expect you to compare some more here with question 2, not only those that I am showing you here. I want you to compare a couple of them. No formal comparison, just like visually compare them. And then 3. Yeah, so that's, this is where you change the magnitude of error, which is the standard deviation. Then you can change the seeds, you can change the associations. And here you come from the for loop. So here you see if you omit confounders and you're using different seeds, to what extent this results in changes in your estimates. And I will keep this part in. Please. Oh, you have followed it. I don't need to do it. This is what is being meant to show to be shown. So I have done it now for you. You just have to go step by step and replicate what I have done here. All right, so this was the lab. This should help you sufficient to complete your lab exercise until next week. So this will be posted on the 13th. So your deadline to complete this is actually the 19th, the Sunday the 19th, which will essentially be Sunday the 19th at midnight. And that should be ample time for you to complete this. All right, good luck with this, and see you next week. Bye-bye.